Well, our next speaker is Emily Martin with the Texas A&M Department of Soil and Crop Sciences Soil and Aquatic Microbiology Lab. She did a very interesting study comparing the three primary methods of E. coli enumerations, and then further looked at those E. coli communities. So, again, maybe not something that we can apply directly to our watersheds, but the more we know and understand about E. coli, the more it helps us to deal with it. So, with that.
Thanks, Kevin. Happy to be here today. My name is Emily Martin again. I work with Dr. Terry Gentry down in the Soil and Crop Science Department at A&M. Our lab is soil and aquatic microbiology, and we do quite a bit of bacterial source tracking in conjunction with the Texas State Soil and Water Conservation Board. And I'm here today to share a little bit of background information and hopefully bring up some maybe questions and considerations when you decide which one of these methods that you may use. So, everybody here in the room today, I've just been for the slide and kind of as an introduction, so to speak. Everybody here is involved in some way, obviously, in looking at water quality and those standards. And to that end, most everybody here, I'm going to assume, is involved probably in some way at looking at monitoring monitoring to look at these standards. So again, what we're looking at now is this indicator bacteria. This whole idea that the standards are based on this indicator bacteria, and that's where we'll spring from today. So kind of another review, like Rocky brought up a minute ago, we start kind of at the top of things. At one point, we were looking at total call forms. Then we were come to find out that those are more widespread in the environment, and so then we narrowed our focus down to fecal coliforms, which are of fecal in nature. And then ultimately, we're down to monitoring for E. coli, which is thought to be the most specific of the indicator organisms. And they're thermotolerant, again, that can run at that 44 degrees C range, and again, is our current standard. So there's two basic ideas here. The difference between membrane filtration and these defined substrate technologies that probably many of you are using here today. So kind of as a show of hands of the ones that are out doing water quality work, how many of you actually use IDEX? And how many folks in the room do membrane filtration? So yeah, so I would say the majority of the hands that went up today are probably using this defined substrate technology. And what's been nice about the use of these newer methods is that they are enzyme-based methods. So we're not just looking at end product production. We're able to increase our specificity with these methods because we're looking at specific enzymes here. So the IDEX technology works off of the cleavage of an enzyme. Beta-galactosidase is the enzyme that all fecal coliforms contain. So if they are able to cleave that enzyme, turns your IDEX wells yellow. And additionally, with the IDEX, you're able to, or the coli alert systems, you're able to at the same time then look at E. coli themselves. And E. coli, the indicator itself, uses an enzyme called beta-glucuronidase. And if they can cleave that enzyme, then we additionally can look at those fluorescent wells. So it's probably a picture that most of you have looked at and counted, and then gone back to your labs and plug in your most probable number table. Where membrane filtration is different. So the wells of the coli alert system is essentially a most probable number technique in that well. So the opposite side of the coin then is to use membrane filtration. So the most common membrane filtration methods out there that many of you that actually use membrane filtration are using this EPA method 1604, which uses a modified Intertech media. Those results there are shown in the picture there on the right. M. coli blue is another one that's maybe lost a little bit of its flair in the last few years due to some issues with their quality control issues from the plant, but there are still quite a few people using it. And then probably one of the newer ones that we're seeing quite some that folks are beginning to pick up using is the EPA method 1604, which uses MI media and can look at both total coliforms and E. coli. The one that we use in our lab for our VST studies and is an EPA-approved method for E. coli is, again, the 1603 method, and it only looks at E. coli. So it's a very specific media there just to look at E. coli itself, where the other two methods have the ability to look at both fecal coliforms and E. coli. So setting up these projects, there's always considerations. And so when folks are beginning to look at these, you have to decide, which one of these groups that you're going to use. And most of you raise your hand for the coli alert system because it's easier. It's a lot less intensive to try to train for. Again, it has minimal training and setup costs, where the membrane filtration does require your lab personnel to have another level of training, so to speak, to be able to conduct those systems. The coli alert system comes pre-packaged. It's easy. It's what we call a shake and bake method. It comes in pre-packaged, where membrane filtration, you do have to create your medias, you have to make your buffers. It is definitely more time consuming in the lab, especially from a QC standpoint as well. The coli alert system, if you have proper dilutions with the coli alert system, your yield, your results are generally very easy to deal with and look at. Some problems that we do have on membrane filtration is overgrowth of these non-target organisms that may preclude us from counting those very easily. But again, as I'll bring up later on in the talk, those non-target organisms are also an issue in coli alert. We just don't see them on a plate. So again, if another consideration from that coli alert system is that if you are going to do any kind of isolations down the road, or you're planning on doing any kind of community analysis from that point on, you're going to have to isolate those from those wells. So that's a whole other step. And if you're doing any kind of confirmatory work, you're going to have to do that anyway. So that adds a whole other layer there. You're going to have to pull those cultures back out of that IDEX well, either replate or refilter to go further. Where if you're on a membrane filter, you can count those organisms that day, and then if you need to do any confirmatory tests, you can immediately put those onto the secondary media and confirm within 24 hours. So just some things to kind of keep in mind. The biggest reason why probably, again, most of you are probably using coli alert systems is, again, the ease of use. So I wanted to show just a little bit of background data from some of the literature that looked at, Ms. Rocky was saying earlier, we've gone from total to fecal, and we've used lots of different methods, and there's been quite a few studies out, looking at, are these numbers comparable across these different methods? Just the count data. This came out of a paper, Hamilton et al. From 2005 that actually came out of Tennessee, looking at direct number comparisons between the MTEC media, coli alert, and M. coli blue. So just to look at the data here, M. coli blue is on the y-axis on A, coli alert data is on the y-axis of C, and on the x-axis on both of these is the MTEC counts. So this is density, this is actual density data, direct comparisons of MTEC versus those two medias. And the R-square values on these are actually pretty good. So you're looking at 84% versus 67% for the coli alert. There is some scatter around this data, but they are generally linear. And so when you're setting up your projects, these are the kind of things that you want to take into consideration, is whether these numbers are comparable or not. So another study that came out in 2000 from Francie and Dorner out of Ohio did a similar study that actually showed even better correlations between modified MTEC versus regular MTEC and coli alert. So back a few years ago, we ended up modifying the MTEC into a thermotolerant organism. So instead of incubating at 35, we bumped up and are now incubating at 44. And so this particular study looked to see if they were comparable across the two different techniques. So the top graph there, again, MTEC numbers on the y-axis, colony counts per 100 mils on the X, coli alert being at the top, again, and then modified MTEC on the bottom. Another study that just came out recently, actually in January of this year, took a different approach. So not only just looking at whether those counts were the same, whether we were able to compare those counts or not, that's from a cultivated aspect. A group out of UC Davis in California went out and looked at independent analysis of these positive wells from coli alert wells. So these were wells that were both fluorescent and yellow, so they were called positive wells. And I really just wanted to show you here, so this is a percentage of clones, so this is just part of the culture independent study. And the two bars, this is a CR is a creek sample, and SEW is a sewage sample. So I highlighted the E. coli in yellow, in red there. So I just, the take-home message from this particular paper is that the coli alert allowed growth of these non-target taxa organisms, and they actually made up a much larger percentage of the community in those wells than did E. coli. Now those would have been positive wells, and there's E. coli there, so it would have been positive per se, but from a community analysis standpoint of what's really in that well, we're having a growth of those non-target taxa. And another thing to note, of note here was in this sewage sample, Klebsiella species were actually enriched. So they looked at these communities. Klebsiella is a known fecal coliform that is a known, that we know creates false positives from a lot from an E. coli standpoint. And at the end of the day, they found anywhere between 14 and 23% false positive rates in their creek water from their coli alert samples. So that's something to keep in mind as you decide what to use. And at the beginning of your projects, it's a good idea to kind of go through and see how, to check your samples for these, just to see how bad your false positive rate may be. So moving on to the study that we actually did in-house at our lab in College Station, where we wanted to look at the diversity of these E. coli isolates obtained from these different medias. A lot of our work is done from a source tracking perspective, and our objective here was to say, if we wanted to do source tracking in more than one watershed across different labs, could we use these, were these methods comparable, and are the communities that we're seeing from those the same? So we took three at the time. There were very standard water quality assessment methods. So we used EPA method 1603 on modified intake, coli blue, and coli alert, not 18, just straight up coli alert, the 24 hour. And we looked at three sampling locations. We just did a surface creek there in College Station, a wastewater treatment plant, effluent there also in College Station, and finally a recreational lake out south of town, north of town there. So again, we took the three water quality assessments, and we went to look at over locations and were the communities that we were able to isolate similar or different across those methods. So we actually pulled 200 isolates per sampling type, per media type, which was quite an undertaking. We confirmed all of those E. coli on mug, again, very similar to the confirmation that's used in coli alert, that same enzyme cleavage that we use to confirm the isolates from the media to make sure that they were E. coli. We used a fingerprinting technique called ERIC PCR, which is a conserved gene across bacteria that we use in source tracking that creates a fingerprint pattern that we visualize on our roast gel. And then we analyzed those groups of fingerprints in a software package called Bionumerics. And we looked at a group or a pattern type at 85% similar, we're called a type, or a fingerprint type. And then we actually looked at the diversity of those types. So just a few results here. So I would like for you to look at the top picture A versus B. So A is an image of fingerprint patterns from the modified intake media, and the bottom is from coli alert. And what I'd like for you to see here is the great diversity of patterns that's in A versus B. You can see that those fingerprint patterns look very similar all the way across from coli alert, whereas you see lots of different types of fingerprint patterns from the intake. So what we saw here is that the intake and the coli blue yielded us much more diverse communities than did the coli alert. So the coli alert isolates that we were able to pull from the well then were almost clonal in nature. So what we were able to cultivate, and just as an aside, we pulled the coli alert media there from there, and then put it on modified intake. We put it through 1603 to get those colonies back out. So what we were able to cultivate then, again, from the coli alert appears to be very much less diverse than what we're seeing with the intake media. Even though they use similar in-goal enzymes to be able to confirm what they are, those two populations on the two different media types were very different. So again, our similarity analysis showed that again. So I've got a figure here showing the overlap between the three different media types. So we've got coli alert here at the top, intake in blue, and in-coli blue there in yellow. So anywhere where they overlap, their Venn diagram there shows what percentage and how many of those actual fingerprint types overlapped. So if you'll notice, we only had 8% of those fingerprint types were overlapped on all three medias. So that's definitely something to keep in mind. That as you move forward, that even though these use similar enzymes and similar methods, so to speak, as to confirm what you have there, and the counts may be comparable, the actual communities that these different medias are selecting for are very different. So as you move forward and you try to decide which one of these methods that you're going to use, I guess I would urge you to think about what you want to do after this. Now, if all you're doing is count data, you're looking for those sort of things. But if you have any goals or aspirations to do anything else further than that, it's definitely something to keep in mind. So again, not only this study, but as well as other studies in the literature have shown that the enumeration results may have some scatter in them, but over time, the enumeration results have been shown to be comparable, but they're not selecting for the same communities. So that this quanti-tray and this MPN technology versus the membrane filter offer these different habitats for growth, and thus different communities are selected. And again, choose the enumeration that's most fitting for the question you're asking in your watershed. So what we're actually going to do with this is expand the geographic, in the future, we're looking to expand the geographic distribution of samples. So we're going to expand this over some different watersheds. And one of the newer EPA methods that folks are beginning to put into their analysis streams is the EPA method 1604 with the I, and we're hoping to include that detects total coliforms and E. coli. We're hoping to include that in our next set of analysis. And kind of as an aside, these medias are very selective, of course, and even though, especially on the modified intake, as well as the coli alert systems are selective, but on a membrane filter, the modified intake is very much more selective, we think, than the coli alert system is. And as far as the counts go, you may see, we're actually seeing in the lab similar to results that what our previous speaker saw, that our numbers from intake, our actual counts off of modified intake, are lower overall than what we're seeing on coli alert. So again, I guess the take-home message would be that different medias select for different communities, and use the enumeration method that you think is best to answer the questions in your watershed. With that, I'll acknowledge again most of our PST funding either comes one way or form or fashion through the state board. Thank them, and I'll take any questions. Yes, ma'am.
Did you do any, this is on raw water, natural water?
Yes, ma'am.
Did you do any on spike samples that were pure cultures?
We did not. We were really looking at, our goal was to look at the communities that we were able to isolate from those natural streams. So we were really looking at not just the numbers per se, is that we wanted to look at, not that we had 10 kids in the room, but what was their makeup? Did they all have red shirts on or not?
On the membrane filter?
Yes, ma'am.
What we've seen when we're doing the global dynamic filter, on some of our columns were turbidity. We just didn't have as much bacteria access to the vagina and food, so that did keep our numbers a little bit lower. So the membrane filter was inhibited by higher solids. Did y'all
We do see that, but honestly, a lot of the watersheds we've been working in the counts, we've been able to look that out. Honestly, we have more, if we do have our main problem with mineral infiltration is this overgrowth, and they don't get those back off of there. As selective as it is, that's been a hurdle.
So I was hoping I could direct this question to TCQ, probably Jill or Joe. I apologize, I did not attend the most recent water quality monitoring and assessment work group meetings you guys just had, just a month or so ago?
The guidance advisory work group meeting?
Yeah.
Yeah.
So I just wondered, I wasn't there, and I just wondered if this general topic, when the bacteria was discussed, got any airtime or deliberation?
I can tell you we used both. It wasn't really discussed as a one result yields different, assessment results, or one method yields different results than the other does, because she kind of said that enumeration results, which is what we're concerned with, are comparable. So we use them interchangeably, and I think a variety of collectors use one or the other method.
Well,
Does that answer your question?
yeah, it does, but I guess I'm wondering if, because of that, there's more grounds for further discussion on that because of the continual issue of, dealing with false positives in these non-critical types of E. coli that need to be somehow weeded out from consideration over time. And I just wondered if it's an ongoing topic that TCQ is interested in or for refining in the future sampling methods or not, or it's just you guys are going to continue to stay the course of both enumeration methods.
We're always interested in the continued discussion, but I was actually happy to see that the enumeration counts are so